Our DNA is like a library with hundreds of books, housed in the nucleus of our cells. Some of these books, known as genes, are vital because they contain the instructions for each and every protein found in the cell. These genes are dispersed throughout 23 pairs of chromosomes, which resemble library bookshelves, because one comes from mom and the other from dad. Chromosomes come in homologous pairs. Different variations of the same genes known as alleles are carried on each chromosome of the pair. DNA is made up of two strands of nucleotides. Therefore, each gene is merely a fragment of this nucleotide sequence. And at molecular level, adenine, guanine, thymine, and cytosine, or e.g. TC, are the four types of nucleotides. Transcription and translation are two aspects of gene expression. The enzyme RNA polymerase uses the gene as a template to build a molecule that can leave the nucleus during transcription. This molecule is known as messenger RNA or mRNA, and it has the same nucleotide sequence as the gene, with one exception. Instead of thymine, it has uracil nucleotides or U. Any three nucleotides in this mRNA molecule or message equate to a specific codon that codes for an amino acid, or a stop codon that signals the protein is complete. The mRNA template used by specialized proteins in the cytoplasm, called ribosomes, to recognize certain codons and match them with the relevant amino acids that will make up the protein during translation. So there are now 64 distinct codons, each of which codes for a particular amino acid, but only 20 amino acids exist, because some amino acids are encoded by several nucleotide triplets. Now, a mutation is merely a change in the nucleotide sequence of one or more genes, but it can also affect vast chromosome segments. These mutations can impact the chromosomes of somatic cells, or any cell in our body other than a gamete, or a gamete chromosomes. Germline mutations are mutations in gametes that can be passed down to the following generation. Mutation can now occur spontaneously or as a result of mutagens. Physical agents such as UV rays and chemicals, as well as biological agents such as viruses, are all mutagens. External variables such as particular chemicals or extreme radiation, as well as internal factors such as an event that disrupts DNA replications and interphase. Mutations frequently develop during DNA replications, which occurs just before a cell divides. During interphase, cells can replicate DNA before they divide. Let's start with a small mutation in a single gene's nucleotide sequence. Three common types include substitution. Substitutions are when one or more nucleotides are swapped or substituted for a different one, such as U for A. Deletions are when one or more nucleotides are deleted, such as U, and insertions are when one or more nucleotides are added, such as A, into the sequence. When it comes to substitutions, the outcome is determined by whether the swap produces a new amino acid, and if so, how that new amino acid impacts the protein's overall folding and function. Now, let's shift gears and look at large-scale mutations, which include having an abnormal number of chromosomes or structurally abnormal chromosomes. These mutations are most commonly connected with genetic abnormalities because they occur as a result of errors during the production of gametes. Let's start with an abnormally large number of chromosomes. If a mutation occurs, we have noted that there are susceptible moments, such as during DNA replication, but there are also vulnerable times, such as during meiosis. Meiosis produces sperm and egg cells with half the number of chromosomes as the organism in fruit flies and other animals. However, such chromosome may not always split entirely. This is referred to as a non-disjunction. This can result in an egg or sperm with an insufficient number of chromosomes. If something goes wrong during the formation of the gametes, they may end up with extra chromosomes or missing chromosomes. We call this aneuploidy. And depending on which chromosomes we have more or less of, aneuploidy can lead to genetic disorders like Turner syndrome, which occurs when a person only has one X chromosome instead of two X chromosome, or an X and a Y chromosome, or a Down syndrome, which occurs when a person has an extra chromosome, 21, making 3 in total, which is why it's also polyploidy, which occurs when a gamete is born with an additional set of chromosomes, is another type of numerical anomaly. For example, an egg may include an extra pair of chromosomes totaling 46 chromosomes, whereas sperm may have only one set of chromosomes totaling 23 chromosomes. When they come together, the consequence is triploidy, which means the result zygote has 3 sets of chromosomes for a total of 69. Tetraploidy refers to the presence of 4 sets of chromosomes, totaling 92. Polyploidy includes triploidy and tetraploidy, both of which are incompatible with life. Finally, consider chromosome structural abnormalities such as deletions, duplications, inversions, and translocations. This can occur as a result of mistakes in gamete production or mutagens such as radiations. Deletions are simple in nature. Essentially, a large portion of the chromosome is removed. A syndrome known as Cry-Duchat or 5p syndrome occurs when a deletion on the short arm of chromosome 5 occurs. Then, there are duplications, which occur when a gamete receives an extra chromosome fragment that may have broken off from another chromosome. The additional chromosome frequently attaches to the homologous chromosome, resulting in duplicate chromosome portions coding for the same genes. Then there are inversions, which occur when a chromosome breaks off and reattaches to the same chromosome, but the process flips it around. If the genes in the chromosome were originally arranged A, B, C, D, they are now arranged A, B, D, C. Finally, there are translocations, which occur when a piece of one chromosome breaks off and is exchanged for a piece of another, non-homologous chromosome, potentially resulting in gene fusion. The Philadelphia chromosome, which is chromosome 22 with a silver of chromosome 9, is a classic example. The BCR gene, which originated on chromosome 22, and the ABL gene, which originated on chromosome 9, fuse right where they meet. So there's a fusion gene called ABL-BCR that causes unregulated cell division, which leads to leukemia. While it's tempting to think of DNA mutations as constantly creating problems, some mutation can actually assist the species as a whole survive by promoting diversity. We wouldn't have any genetic diversity or the ability to adapt through time if they didn't exist. In fact, every characteristic that distinguishes one person from another can be traced back to a genetic mutation.